Hello, and a very warm welcome to our Cell and Gene Therapy Insights webinar on AAV manufacturing process development using fast upstream and downstream HPLC analytics. I'm Charlotte Barker, an editor at BioInsights, and joining me today is Alice Strancar, Executive Managing Director at BIA Separations. Alice is one of the main inventors of convective interaction media monolith columns and co-inventor of many analytical methods and purification processes, including ones for pDNA, mRNA, AAV, and viruses. Alice is co-author or author of more than 90 scientific papers dealing with separation and purification technologies. Today, Alice will talk about a fast, at-line HPLC-based system that enables analysis of the full and empty capsids ratio directly in the harvest, as well as giving insights into purification. First, we'll have a presentation from Alice, followed by a Q&A discussion. As always, do feel free to ask a question at any time throughout the event using the Ask a Question box, and we'll try to get to it during the session. Before we get started, I'd like to draw your attention to the Resources tab, where you'll find links to get further information on the topics discussed today. And with that, I'll hand over to Alice for today's presentation.
Thank you, Charlotte, for the introduction, and hello to everybody. So let's move to the presentation. So the work I'm going to present is work of quite a number of my colleagues in the company, and with this I would like also to thank to all of their hard work and input. And before moving to the topics, just shortly presenting who we are. BIA Separation has been acquired by Sartorius end of the year 2000, so we are under Sartorius umbrella now for more than a year. Since the very beginning, more than 22 years ago, we directed our efforts to serve the gene therapy market, which means we were looking for better suited products and methods to deal with viral particles, plasmid DNA, recently also mRNA. We are also now Center of Excellence of Sartorius for these efforts, and still our main occupation, our main task would be to develop new products, especially new chromatographic columns based on the monolithic supports. Within last 20-25 years, we have been helping customers. We have been developing for general use a number of different processes, which includes plasmid DNA. Nowadays, this would work with small plasmids, mid-sized plasmids, and very big plasmids. Even more than 30 kilobase per plasmids are possible to purify with our platform process. We have quite some experience also with other types of DNA, like minicircle, doggy bone. Recently, much more efforts we directed towards mRNA, first oncolytic mRNA vaccine, and recently corona. We work with adenovirus vector for more than 20 years, and also with adeno-associated viral vectors. We were developing more than 20 processes or part of the processes dealing with AAV purification, and also some other viral particles like influenza, vaccinia. Lately, a lot of work with exosomes, phages, bacteriophages, so viruses which kill bacteria, and also other viral-like particles. This is more or less our main target. We have also some experience with some proteins, especially from human plasma, but mostly we deal with viral particles, plasmids, and mRNA. I would say it's probably useful for you to understand the way we are working. We always try to serve our clients. The needs of our clients is in as fast manner as possible, because we know that many of you would be looking for moving fast to the next clinical trial projects and towards the market, and it is very important that you have support which you need to reach your goals. And during this time, we were serving quite a number of clients. Some of them were willing to share some quotes, like colleagues from Avexis. We were helping Avexis to bring their Zolgesma to the market. The process was really developed from centrifuge process to fully scalable chromatography-based process just in about 1 year. When there is a need for new ligands on chromatography column, we can also serve our clients very fast. Just within 6 months, we are delivering to Arcturus oligo-DT column, so first prototype and CGMP compliant column, just 6 months time in between. And when it comes to the tech transfer of processes which develop or we co-developed, we can also act very fast. In March last year, we struck a deal with Bionet to develop the manufacturing process for mRNA for COVID. In October, the first batch for clinical phase 1 trial was produced. So the complete plasmid DNA purification, IVT synthesis, and also purification of mRNA, all developed just within 6 months and tech transferred to the Bionet Asia production facility. Why we are able to serve our clients so fast? First of all, there is more than 20 years of experience of a big group of researchers, developers in our company. More than 60 people are working with method development. The second reason is due to the properties of our chromatographic media, which are unique on the market. They are known to be continuous bits with continuous channels, which are not discontinued. So we would have no diffusivity constraints in such a chromatographic support, neither shear forces, because the channels would allow for laminar flow, which means no shears, no eddies. And secondly, we have no detent pore, which means the mass transport between stationary and mobile phase is instant. So we can speed our processes to be really extremely fast. The equipment is actually the limiting factor here. In addition, the properties of monoliths would be flow-independent resolution and capacity, which allows us to use extremely high volumetric throughputs at laboratory scale, like maybe 20 column volume per minute flow rates, which means 20 ml using 1 ml column. These high flow rates may not be feasible at industrial scale because with 8 liter column, this would mean 160 liter per minute. But due to the flow independent resolution and capacity, you can nicely lower down the volumetric throughput by even an order of magnitude, and this would have just no influence on the resolution and capacity. Due to the easy access of very big molecules, viral particles, to reach the surface, we can provide much better capacity for viral particles when compared to the resin based on porous particles. It can be even 2 orders of magnitude higher capacity, which means instead of using 100 liter column packed with porous particles, you can shrink down the size of your column to just 1 liter. Due to no shear forces inside the monolithic structure, you can count on better recovery of your target product. Like with plasmid DNA, especially big ones, which are shear sensitive, you can usually improve the recovery by 10 or even more %. And due to no back mixing, due to no diffusivity, you can also count on better resolution, which actually helps to separate empty viral capsid from the full viral capsid. Other than that, this is just chromatography resin, so any ligand you can imagine on porous particles, you can also realize on the monolith, from cation exchangers to anion exchangers, hydrophobic interactions, affinity interactions, and so on. Yet another product from our company would be HPLC. we are naming the HPLC PATfix because we wanted to emphasize its main use. We are using this fast HPLC system to speed up development of our processes, to help our customers so they can speed up their processes. And what is so unique with our HPLC system? First of all, the pump heads we are using allows for using sodium hydroxide 1 molar for a long time without any leaking. Also, high salt concentration can be used again without leaking. Even more important would be that we allow for connection of detectors from different vendors. So our software is open to connect detectors from different vendors, which allows us to connect in line, in series, UV detector, a fluorescent detector, multi-lysis scattering detector, refractive index detector, and so on. The system also allows to be easy turned to semi-prep. We just unscrew the pump heads with 10 ml flow rate, and we screw in 50 ml pump heads, and you have semi-prep system with all the same detectors and everything, so you can basically do first scale up using the same system. When it comes to the methods, we are aiming to provide system with already built methods. The first such system would be plasmid DNA, PATfix. So this would have the method build it in with a validated plasmid DNA method. The method can be used either to check for the quantity and the purity of the final plasmid DNA product, or can be used as an in-process control to optimize your purification plasmid DNA method. The next one to come in a month, it's mRNA-based method. So there will be actually 4 different methods built into the system, not only to optimize the synthesis of mRNA and quantifying mRNA, but also to quantify for a double-stranded RNA, quantifying of residual proteins. And then the next one to come would be the AAV system. For all this, we also provide special columns optimized for the task, like plasmid DNA column, mRNA column, Prima S, which basically allows you to use the system and the method, just do revalidation, and you are ready to go. You can save a lot of time not looking for columns, not looking for development of the methods. As already mentioned, the columns can be, analytical columns can be of any traditional chemistry, but we also have dedicated columns like for plasmid DNA. Also in our analytical columns, there would be monolith in, so again, just channels inside of the resin, which speed up your process. Very important for analytics, it lowers down the carryover, especially of molecules which tend to be entrapped into the dead-end pores of porous particles. Important to say, the connectors are such that they would allow you connect the system to just any HPLC system, which means you don't really need our HPLC system to run this separation, but our HPLC system would allow you for better flexibility and better sensitivity. And now the most important part of the system, the possibility to integrate different detectors in, and here is basically the example of this. So on the left side we see the picture of the same chromatogram as on the right side. Just on the left side we have UV and multi-light scattering detector. On the right chromatogram we see fluorescent detector and multi-light scattering detector. So on the left side, the red color represents the UV to 60, the blue UV to 80 nanometer, and the black represent multi-light scattering detector. When we get the sample, a known sample from our customer, we would, if it's AAV, usually run this sample on the cation exchanger in the linear gradient, so we would get quite a number of peaks, and we would not know which of these peaks would be the AAV, the product. So if you have Mal's detector, usually the biggest Mal's signal would be due to the AAV scattering. So we can recognize easily which of those peaks in the UV signal would be our product. We can start even to quantify our AAV directly in the harvest, and this can serve us also to calculate mass balance of our AAV product, so we can easier define the yield of our process. Even more important, on the right side, we can see, in addition to the Mal signal, we can see the two signals from fluorescent detector. We are using dual channel fluorescent detector. The first one, which is orange type, would be tryptophan excitation, so we can boost the sensitivity for host cell proteins. And more important, by using fluorescent on the picogreen, we can boost the signal or the sensitivity for DNA-related impurities, which may carry also host cell DNA or part of it. And we need to pay attention that this is not really going to our final product because it may be really problematic. How we can realize this analytics, because the DNA is not fluorescent, we put some picogreen to the sample before injecting, and picogreen would stick to just any DNA-related impurity, and we would then get a signal for all of DNA-related impurities. And then we can also look how we are cutting down the amount of these DNA-related impurities during our purification process. Yet another orthogonal method would be ultracentrifuge. Actually, ultracentrifuge is routinely used for many decades, so there are a lot of data also on the ultracentrifuge, but usually we would take pictures. We wanted to have a bit more data from the ultracentrifuge. We wanted to have orthogonal method to our HPLC, also orthogonal to the PCR, and we basically designed kind of a holder which allows us to pump the material from the ultracentrifuge after centrifugation through a small capillary, HPLC capillary, directly through the detectors, HPLC detectors. So whatever is on the bottom, when pushing from top to bottom, would go through the capillary out and would go directly to the HPLC detectors. So we bypass the pump, we bypass the column. And if we now check for the let's say for the usage of this approach, we usually would start actually in our development this way. We would pre-purify the AAV on the cation exchanger. We would take part of this fraction and run the ultracentrifuge. And you see on the right chromatogram down that whatever comes out at the very beginning would be the most dense stuff. So we start from the right to the left, again with the same detectors. So blue to 80, red to 60, fluorescent orange, and multi-light scattering detector black. And we see that there is some dense impurity, and then we get the full AAV where red is higher than blue, and in the end, on the top of centrifuge or at the end of this centrifugram, we can see the empty capsid. So we would have a clear idea about the ratio empty-full. Also, if there are some other species but the full. And with this in mind, or with this picture, we would then go and try to develop our chromatographic method to reach such kind of separation or purity. So centrifuge is always much faster to separate empty and full, but when you need to do the scale up, you need to invest time and efforts to improve the separation method on the HPLC so you can reach the separation which is needed to provide you, I don't know, with 80-90% full. If you just inject on the column, the sample, you would usually not see immediately the separation, like you can see here on the left, that the full and the empty are quite together. So only with improvement of the buffers, with the pH, and so on, you can get better separation, or maybe you even need to go to another ligand. And then when we start to develop our processes, it is very important also to take into account that USP, so the production of the particle in the cell line and the purification of the virus particle should be regarded as one process. Otherwise, we would have quite some issues with the scale up. We would need to return again, back again and again, and reshape the purification to satisfy for the robustness of the process. So again, we need good analytics to connect everything together, and HPLC is one of the methods which can be orthogonal to other methods like PCR to give us much better insight in the quality of our upstream, in the improvement of our upstream, in the robustness of our upstream, and also in the improvement of the expression of our product. So how to realize this? We would deal with a sample with supernatant, which is very complex. So what we build it is kind of a method which allows us, in addition to the PCR as orthogonal, also to check for the empty and the full virus particle. So how this can be realized? Quite simple, actually. We take the supernatant, we filter the supernatant, and we inject everything to the first chromatographic column, which is cation exchanger. We could see that this one basically can bind and dilute any AAV serotype. We would do the pre-purification and concentration of our AAV on this column, and the fraction which contains the AAV. We would, by switching method with switchers, inject to the second column, which is the column which can separate the empty and the full particles. And the detection would then be by multi-light scattering detector, which is most selective one, and this would allow us to not only see the amount of the full particle, but also the ratio between full and empty particles. How this can be realized? We perform pH gradient to elute from the cation exchanger and bring the pH up to bind the AAV on the anion exchanger. Always important, and I'm repeating this again and again, if you don't use orthogonal methods, we may cause quite some mistakes in our predictions, in our analytics. So we were checking the accuracy of our HPLC method with the PCR, and here on the right up chromatogram, you just see the zoom in of different steps in our upstream. So the transfection starts with the orange, and then after some time we take the sample, and you see that only empties are formed during so more time of the process. Then the fulls are starting to be formed. At some point, actually, the empties and the full would not grow anymore. We check then at the end also by lysis of the cells if the ratio is similar in the cell as it is in the supernatant, which basically confirms it is. So we can kind of conclude that the ratio of empty and full would be represented by supernatant. But this may not be the case with the concentration of them, because if you compare everything with the PCR, we would see that in the supernatant we only see part of our product. PCR would detect everything, but the HPLC would not really give us the amount which is in the cell. Of course, if the amount of AAV in the cell is different than in supernatant, we may not have the data which is accurate, but at this moment this is not even that important. More important is that we have a method which basically allows for the analytics of the ratio between empty and the full. And once we have such a method, we can start to think about optimization of our upstream, which is actually the case in this slide. So we're interested to check different transfection reagents, how they operate with regards to the expression of AAV, and also with regards to the empty and full formation. So we mix transfection reagents and plasmids in different transfection reagents, and we then transfect. After 72 hours, we took the samples, and different colors represent different types of transfection reagents. If we go to the next slide, we would then see the results more in detail. So multi-light scattering detector zooming chromatogram on the right with different colors. Each of these colors represent one of the experiment. So the blue color represents transfection reagent 1, also with the bars showing also what was the ratio between the DNA and the cell amount, and also transfection reagents and the DNA amount. So we see here from this experiment, we see that transfection reagent 1 and transfection reagent 3, which is the last one, are basically quite overlapping with regard to the amount of full, and also with the amount or the ratio between empty and full. But all other examples, either due to different ratio between transfection reagent and the DNA, or due to the different transfection reagents type, would not really result in formation of the fulls. We can get quite a number of empties, but almost no full. So it is really good to have such a method to allow us for optimization of the upstream. And once again, in addition to the MALS HPLC, we would be using PCR, so we can double check in the orthogonal way if our results are correct, which is also showing here in this graph. So on the left you see the PCR results with bars. Different color means different experiment, and on the right you see the amount which was defined, or which was extracted out of the HPLC data. And once again, using such a method, we could conclude that transfection reagent 1 and transfection reagent 3 would be good enough to proceed. The next step might be to check for the mixing of our upstream, or temperature, or the fit. So we have a possibility to really optimize our upstream relatively fast, not waiting for the downstream results, or waiting until everything is done and you perform on analytics. Of course, it is also important to have analytical methods which can help you with understanding of key impurities, and also help you with the downstream process development. Our aim when performing downstream development is always to remove the most problematic impurities, which usually would be connected with chromatin structures, or would be connected with nucleosome structures. So nucleosome is a aggregate or complex between the histone proteins, so highly positively charged proteins, and DNA or RNA, or both, means highly negatively charged nucleic acids. Small nucleosome can be just of some nanometers, but when they start to grow, they get kind of a chromatin structure, so much more complex structure of any size, of any shape, and also of different charges. Really very much depends. If we have more histones on the surface, we would have more positively charged chromatin. If we have more DNA on the surface, it would be more negatively charged on the surface. And these aggregates, these complexes, can be even bigger than half micrometer So some of them would be the size of our viral particle, and they might collude with our viral particle depending on interactions. And we also need to take into account that histones are very hydrophobic, so they may also stick to the column and would not be that easy to wash them out. So really structures which are very important to watch on if you would like to have safe product, because they would be carrying host cell proteins and host cell DNA through the process to the final product if we don't remove them properly. We can think about DNA treatment. We can partially remove these structures, but we need to keep in mind that when the DNA is packed inside of such a complex, the enzyme is excluded. No chance for the enzyme to digest such a chromatin. It could be even worse. Even the primer might not be for the primer might not be possible to multiply our residual genomic DNA, and we might think we have no residual genomic DNA in our sample. So we also need to pay attention on this. And as already mentioned, if we put picogreen would be would be possible to penetrate inside of such complex, and we can detect such a DNA. Not maybe possible to be detected by PCR. We can detect it with picogreen fluorescent detection. So having clear idea how to move ahead and analytics in place, and here is one of such analytical chromatograms. The DNA is on this chromatogram presented in with a green curve on the right, which is zooming of the left chromatogram. So the chromatography is size exclusion. We are using the exclusion pores of 4,000 angstroms, and most of the chromatins would come in the void volume, but there are also some chromatins which a little later on, nucleosomes are even smaller. So we would have basically these chromatin nucleosome structures over all the size exclusion chromatogram. But once again, more important is we have a method which allows us just within 30 minutes to get an idea about the amount of chromatin structures. And then with these methods in place, we can start to really think how to develop the process. When it comes to the AAV, if the AAV is not bursted out of the cell, we need to lyse the cell. Usually two possibilities would exist, either detergent or salt lysis. We try to avoid detergent because scaling up with detergent might be a bit of an issue. Micelles are formed and scalability is not very straightforward. With salt you would have less issues, but if you use the salt, then you need to use salt tolerant enzyme. We have a product for this, which we call it kryptonase. So this is salt. So this is an enzyme which is able to digest DNA at high salt concentration, like 0.5, 0.6 molar sodium chloride. In addition to this, we would also use one of the methods to remove chromatin. This can be flocculation, can be TFF, can be solid phase extraction. But if the ratio between the product, so if we express our product with very high titer, meaning like E11 particles per ml of our harvest, then we may just go directly to the cation exchanger or to the affinity column to do the purification, and then the polishing and improving the ratio empty full can be performed by using either strong anion exchanger QA or two multimodal ligands, which is Prima S and Prima T. Both are weak anion exchangers with some very edge bond characteristics, and this also proves to provide a good alternative to the strong anion exchanger. Actually, for some serotype, this basically works. This may work better than the QA. Just as one of example of chromatin removal would be using CIMasphere. These are kind of special particles which we just put into the harvest, and they would stick to the chromatins and would usually remove 90 to 95% of chromatins. which basically then allows to go for the next step. So after this filtration of these particles out, we would go to the cation exchange column. There are still some chromatins, small ones, eluting before our AAV product. If you look on the purity of this AAV in the fraction E2, there is still a small amount of H1 histone protein, which means that it is still not full separation of chromatins. As I mentioned, they are very sticky. So to really have extremely good removal of chromatin structures, the best would be to use very strong cation and very strong anion exchanger. CAP peak at the end, very big, shows that although we are using the solid phase extraction of chromatins, we still have quite some chromatins present. So really something to keep an eye on all the time. And then we take the fraction of the AAV and we go, in this case, to the QA, where we can separate full and empty. And we see still also on the right the CIP peak, still a small chromatin peak. Some of the chromatins were of the other charge, opposite charge, and they were sticking now to the anion exchanger. And now the method to measure the chromatin removal. So as already notified before, the green color means fluorescent picogreen detection, and all the area which is now green would be related to chromatins or very small fragments of the DNA. After the solid phase extraction with CIMasphere on the right top, we see that most of the green color is disappearing, so we were already removing 90–95% of the chromatins. Then cation exchanger, really working course for most of the residual chromatins removal, so the green goes even further down. We still see small amount in the void volume, small amount of chromatins at about minute 10, and this would then be further removed with the strong anion exchanger, and also the ratio to 60:80 is improving. And now to the last presentation, or to the last topic of today's presentation, and this is how to improve the separation of empty and full. This separation is really tough. We are dealing with two capsids which are very much similar, but it is known that they have different behavior when we are working with some cations or anions. So if we are using bivalent cations, empties would usually be stabilized. Urea is also having influence. EDTA, because it is influencing or EDTA is complexing our cations. Keep in mind on the citric buffer, which in some cases is used to elute from the affinity column. So citric buffer is making complexes, or it is getting into the complex with magnesium or precipitate magnesium or calcium. So all in all, magnesium has quite some influence on the stability of empty capsids. If you would have at least 2 millimolar magnesium in the buffer A and buffer B, all the empty capsids would elute in one single peak, as it is shown here on the left chromatogram. So to the blue to 80, higher than red to 60, and the right is the full capsid. Again, 3 different detectors. The chromatogram on the top is UV, the middle one is fluorescent tryptophan, and the bottom is multi-light scattering, the black. If you would have no magnesium in our buffers, and there would be no magnesium in our sample, we would see at least part of empties to stick to the column, to the QA, to the strong anion exchanger, more than other part of empties. And this can now be seen here on the chromatogram, on the right chromatogram, on the top. So 260 to 80, blue to 80 higher than to 60, still part of empties. Then the full pool is never moving. It is always on the same position with or without magnesium. And then there is a third peak coming here. And these are other empties which, due to the no magnesium, change their charge on the surface and stick to the QA columns stronger. Such kind of separation without magnesium is not good for analytics because we would need to quantify several peaks, but it is definitely good for the prep, for purification, because this part of empties are nicer removed, or there is better resolution between the full and these empties, which I look later on, than when we use magnesium, because the empties and full are really closely together. More important, the magnesium amount was giving us kind of thinking how to improve the separation of empty and full by designing such a ligand. So by designing a ligand which would use the properties of magnesium or calcium to further separate empty and full. And one of such ligands is actually PRIMA-T. And we can see here in this chromatogram that we are really able to separate full baseline from the empty. The separation is done in magnesium gradient. We run gradient from buffer A to 50 millimolar magnesium chloride as buffer B with a very shallow gradient, and we see not only the full, but there is something which is kind of a tailing. But for empties, we need 200 millimolar sodium chloride, so most of empties are really sticking to this column very well and can be very well separated from our full. But let's see now the zoom of this full. So here we see on the top, we see the zoom. And on the left chromatogram, on the top, we see that basically this full is not kind of very homogeneous stuff. We still see some small peak on the left where the blue is higher than the red, which means we still have some empties. Then there is a main peak where 260 to 80 really represents the full. And then there is a tailing, or actually a peak on the tailing, which represents something else, probably not our product. And we took this fraction and we put it in on the centrifuge. We perform centrifugram, and we indeed see also on the centrifuge that we have two populations of full and still some empties in. But the most important are these two populations of the full. So with the fluorescent detector orange, higher peaks, and then 260 to 80, red and blue. And we can see that at the beginning of the peak, the ratio to 60 to 80 is higher, but on the tailing, the 260 ratio is dropping. So the question here would be, do we have a shorter DNA inside of this AAV? We were not really inspecting what's going on, but we believe that probably this part is not good to have, and it is not really easy to separate this stuff. So our way to deal with this would be go back and optimize the upstream to result in this second peak, because this second peak would be really difficult to separate from our product. But at least we have an analytical method to deal with. So to conclude, very much again and again kind of calling for orthogonal methods. We need more methods. We definitely need still more analytics to better understand our gene therapy products. Ultracentrifugation and chromatography are nice orthogonal methods, and by using special detector setup, we can get more data, more visibility of things. PATfix system, the HPLC system, allows very different approaches, very different analytics. From one side, multi-detector use, and on the other side also using switching methods, and soon also inline cell lysis, so we would be able to also lyse the cell in the system itself. When we are designing the manufacturing process, we really need to pay attention on chromatin, because chromatin would bring impurities which may be dangerous. to our final product, especially because we may not see them. PCR may not detect them. It is the picogreen method with size exclusion column or any other column which improves the sensitivity for residual chromatins. And we need new columns, we need new chemistries, new ligands to even have better separation of empty and full, not really empty and full, but also of either truncated or however we call the capsids, which are not really our product. With this, I would like to thank to your attention, and I'm happy to go through the questions which you may have during the presentation. So thank you very much.
Thank you, Alice. Great presentation. So we are going to move on to the Q&A now. Do keep the questions coming in, and we will try to, any that we don't get through today, we'll try and answer by email, but we've got about a quarter of an hour, so we should get through a few. Okay, so first question I've got here, what kind of sample pre-treatment do you need to inject the crude bioreactor sample into the PATfix?
We just filter stuff through the 0.2 micrometer filter and we inject directly.
Brilliant. And what is the delivery time of your columns and your PATfix system?
Some columns in principle should be on stock, except the biggest industrial one. This may need up to 6 weeks. The PATfix system also depends on the configuration, either mass detector or not, but usually it will be up to 2 months.
Great, thank you. And which moles detector are you using?
We are now basically OEMing mass detector, so it's kind of OEM from one of the vendors. It is more sensitive than the one we used in the past. In the past, we were using wired detectors. It is more sensitive and cheaper, so we are trying to bring this multi-light scattering detection to our clients, I would say, with more of affordable price.
Thank you. What would be the price of a PATfix system to perform empty-full analytics in supernatant?
Yeah, for this we would really suggest to go also with the mass detector, so the full setup of detectors with auto-sampler and switching techniques would be at around 150,000 euro.
Brilliant. And can you analyze the empty-full ratio in the cell?
Yes, but only after you do cell lysis. At the moment we do this offline, but we are developing inline lysis method, so we hope to be able to provide the PATfix system such that one can perform inline lysis and go directly then to the column.
Thank you. Would your in-process HPLC work with any AAV serotype?
Yes, not only the system, but all our columns are also suitable just for any serotype. So far, we had in our hands more than 20 different AAVs, not only known serotypes, but also quite a number of chimera. And cation exchanger is basically working just with any serotype. When it comes to the anion exchanger or to separation of empty and full, then you need to go and select which of the three would work for your serotype the best.
That's great. And a follow-up question from that. Is the strong exchange empty full column good for any AAV serotype?
No. Like AAV9 with the strong anion exchanger, you would probably not be able to do the separation. There are differences from within the same serotype, either you see more glycosylation or less glycosylation, which results in wider or narrower peaks, but usually the strong anion exchanger would not provide you with empty full separation for the AAV9.
Thank you. And are your analytical empty full columns scalable?
Analytical columns are basically optimized for the best sensitivity, for the best resolution, not necessarily for the best capacity. So they are a bit different than our preparative column. The The separation which you see on the analytics would be similar to the prep columns, but not the same. So if you would like to do the scale up or scale down of your industrial scale process, you should basically use 1 ml column, which is then scalable. There is another difference as well. Analytical columns are axial columns, and the preparative columns are radial columns. So there is another also issue. So you need to jump from axial to radial if you go from analytics. So analytical column can be a good guidance, but it is not directly scalable.
That's great. We're getting lots of questions coming in. How variable are the results of AAV purification using HPLC depending on AAV serotype?
I don't think it's so much about serotype. It's more about the purity and also the quality of the harvest. We basically, with the harvest material, so if we basically go with the harvest directly to the column, we can expect plus minus 10% variation in the results. With more purity, we should get down to plus minus 2%.
Brilliant. And a follow-up question: would this approach work with a shuffled AAV capsid library?
Yeah, I believe yes. I believe yes. I have no clue about the amount you are using to basically work with the library. We would need to think also about the amount which you need to inject, but in principle, yes.
Yeah. And what is the picogreen incorporation success rate for the DNA level?
We didn't really check for this kind of to really check for every chromatin structure. Usually, whatever has the DNA would be sticking with picogreen, but of course, the amount of picogreen may be different from the length of the DNA. So it is not kind of linear. We cannot say that whatever we see under this chromatogram or under this huge area would be linear. So linearity is not existing unless you have just one single size of the DNA.
Absolutely. We've got a viewer here with 3 questions, so really grilling you. So the first one, how do you integrate the questions are regarding the HPLC method for empty-full ratio determination. First question is, how do you integrate the peaks as it's not well separated?
Yeah, this is also a good question. It would be depending. If you have 2 peaks which are of quite the same size, we then just do the valley down to the baseline. But if either full or empty is much smaller, in a much smaller amount, then we would basically superimpose the peak and integrate as a superimposed peak. Also, it's very much important which detector are you using. We found out that the fluorescent detector is the most accurate when it comes to the quantification of empty and full of the pure sample. If the sample is very, very, I would say, complex, like in the harvest, then Mal's detector would be the best.
Absolutely. Well, I think you've answered one of the other questions there, but one more question. Do you think it may be possible to improve the sensitivity by adding a fluorophore?
Yes, sure. If you find a good one, you can definitely improve the sensitivity.
Brilliant. Great question. Thank you, viewer. So another question here. You had mentioned removing second peak, empty capsids, or eliminating it altogether by working on the upstream process. What are some of the parameters that you examine to generate more full capsids in the upstream process?
There are several basically factors which can improve or actually work to the opposite direction. The ratio between the transfection reagents and plasmids is important. The type of transfection reagent is also important, but it is also important to work with, let's say, good plasmid, means plasmids of a good purity, If plasmids are not pure enough, then you may see quite some issues. In some cases, it is maybe even good to not use 3 plasmids, but maybe 2 plasmids and 1 doggy bone. So there are many different possibilities to improve the quality of your expression.
Absolutely. Can you elaborate on the difference between CIMac QA and CIMac AAV full empty columns, as they share the same ligand?
It is just as I mentioned already at the beginning. There is a bit different ligand density. There is an axial column in comparison to the radial. So the idea of our analytical column is to have the best possible resolution, and of the prep is to have also good capacity. So our analytical columns are not really optimized for capacity, but for resolution. One of the key difference is the ligand density, and the other one is in also the way how we operate the column. So analytical is axial and prep is radial.
Brilliant. And I think this viewer is asking what's the time until you get your results? What's the time to result assay?
If this is a question for AAV switching method, this is about 30 minutes, and you can basically, you then maybe need some time to integrate, or if you trust to integration of the method, it would be 30-40 minutes, and you have the result.
Brilliant. And what is the sensitivity for impurities levels?
Now very much depends on which impurity. With picogreen, sometimes we can reach sensitivity which is even better than the PCR, and this is, as I mentioned, sometimes PCR primer is just too big to kind of accommodate the multiplication of the digested, or let's say part of the DNA, which is inside of the chromatins. And basically this is usually our kind of main goal. I can not say numbers, I don't have numbers in my head, but it would be in some cases better sensitivity for the residual DNA than the PCR.
Brilliant. I'm sure we can follow up later with more precise numbers as well. Can these analytical columns be reused, and if yes, how many times?
Yes, of course. The analytical columns should survive for at least one experiment. Otherwise, makes no sense, because if you need to remove column for each experiment, leads nowhere. So we perform after each round. We perform washing of the columns. This is why also the method is longer. But it is really necessary to wash the column after each injection, and if this is done properly, and if the upstream is not really nasty, then these columns would perform at least 30-40 runs, sometimes can be even a few hundred times.
Brilliant. Well, thank you, Alish, for answering those questions. I think that's all we've got time for today. Any questions that we didn't get to, we'll try to reply to those by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. So I think then all that's left is to thank Alish once more for a great presentation, and thank you to the audience for listening. We hope you'll join us again soon.
Thank you
Goodbye.
to everybody to take time, and thanks to you to lead the conversation. Bye-bye.